Hello, everyone. So today we'll be getting into the immune system. Immune system is a collection of cells and organs that are responsible for the ability to resist infection and disease. So I think a good way of just describing it is to go against bacteria, pathogens, and viruses. I guess that's a good way of grouping it. And there's components of it in basically every tissue of the body. Now, the body has multiple layers of defense against infection. The first is using physical barriers, and the other is using cell-mediated immune responses. Using physical barriers are created through non-immune cells. Non. And this could lead to structural features which prevent a pathogen from entering the body. This includes such as skin or hair, but it also includes chemical secretions, such as stomach acid or lysosomes that can be used to neutralize and destroy pathogens. The integument is the most important physical barrier of the body. The secretions from the sebaceous glands and sweat glands can wash away potential pathogens. The hair keeps potential hazards away. And there are the epithelium being stratified squamous can be interlocked, keeping intruders out. And the mucous membrane also contains multiple defenses. The mucous membrane lines the digestive, respiratory, urinary, and reproductive tracts and provide protection against foreign substances and bacteria through their secretions. Now, responses are determined through white blood cells. White blood cells, also known as leukocytes. White blood cells are found in many places, but they're all born from the bone marrow. Red bone marrow. However, they have differentiation patterns where some of them can be lymphocytes. Well, some they can be from myeloids, but they can also be from lymphoid stem cells. If it's from lymphoid stem cells, you're going to go to, so let's say it's from lymphoid progenitors, it's going to become a lymphocyte. Whereas if it's a myeloid progenitor, it's going to be a monocyte, neutrophil, eosinophil, or basophil. And immune responses involve multiple components. One is detection of invaders, secretion of chemicals and lastly, destruction. Some white blood cells participate in innate immune responses, whereas others are part of adaptive. Adaptive include T and B lymphocytes, whereas everything else is part of the innate immune system. Now, the lymphatic system also consists of vessels that, well, on a separate system. The lymphatic system contains vessels that move lymph throughout the body, and they can protect or produce or house lymphocytes. Imagine just like the body has another circulatory system full of lymph vessels, lymph, and it resembles blood, lacks cells and plasma proteins. Its only function is to produce or house lymphocytes. The lymphatic system is non-synonymous with the immune system, also has non-immune functions, and one of the functions is to return filtered fluid from interstitial fluid to plasma, and the second is transport of transport of digested and absorbed lipids. They also have differences from blood vessels. For one, well, I guess similarities. They all have an endothelial wall around lumen. And for two, uh, larger lymph vessels have a smooth muscle layer and valves. Differences: lymph capillaries have blind ends. They're more permeable around their length, and there's no equivalent of a heart. Instead, they're reliant on smooth muscle contractions. So the big idea is, okay, similar to blood vessels, you have an endothelial wall around the lumen. The larger ones have essentially valves and smooth muscle layers, but the difference is contraction is done using smooth muscle rather than a heart. They're more permeable and they also have blind ends. I think the blind ends should also be another structural feature that is pretty key and something that is definitely something you should notice, I suppose. There's also the lymphatic system also composes of lymphoids, lymphocytes, lymphodes, and lymphatic tissues and organs. Tissues have lack a capsule, whereas organs have distinct capsule. Now you can have primary and secondary. Primary organs, lymphatic tissues include bone marrow. Primary organs include the thymus. Secondary organs can include lymphoid tissue, primary tissue, and secondary organs include the lymph nodes. So secondary tissue contains mature and functional lymphoids, whereas primary is to the production and maturation of lymphoids. The lymphatic system is not synonymous with the immune system. Not all white blood cells are lymphoids. Only lymphocytes are lymphoids, come from lymphoid stem cells, while all the other white blood cells are from myeloid stem cells. But not all lymphocytes are associated with adaptive immune responses. For example, lymphocytes, T cell, B cells, these are all lymphocytes, but these two are part of adaptive, whereas natural killer cells are part of the innate. Lymph nodes are in-housed, are also lymph nodes are particles that basically connect through and house mature lymphocytes. Lymph nodes can also monitor lymphs, monitor for antigens in lymph to stimulate immune responses. Imagine these like what's it called? What's it called? Firefighting halls? Firefighter uh like fire halls, right? Where you would have firefighters and stuff go through them. They can signal antigens, meaning, oh, they can signal there's a fire somewhere, and they also are for training purposes. They are able to store and mature and allow lymphocyte maturation. All big concerns are all big functions. They filter lymph and they act as posts for generating adaptive immune system responses to all that and whatnot. Antigens enter the lymph node through afferent vessels, and yeah, just I guess there's an order we can talk about. Uh, Afferent lymphatics. The order is they first go from the afferent for lymph, it enters from the afferent lymphatics to the subcapsular space to outer cortex to the paracortex. Then it goes into the medullary sinus and then the efferent lymphatics. And then lymph leaving has been filtered. It's been filtered Active lymphocytes return to circulation through this lymph, and these activated lymphocytes have acquired specific adaptive immune responses that leave and they leave through the efferent vessels. Macrophages in the medulla help destroy pathogens that make it through the lymph vessels. So I think I should label this. Um, in medulla, destroy before reaching. So in a way, it's also a passageway for a lot of pathogens to at the well, you have blood circulating in well, you have lymph fluid circulating in it. So you have a lot of pathogens that are entering, exiting this way and having at least some responses. Again, lymph nodes monitor lymph for antigens, stimulating responses. And not all mature lymphocytes are associated with lymph nodes. There are also secondary lymphoid tissues, such as the spleen, malt, or lymph nodules, or aggregations of lymphocytes. Lymphoid nodules are regions of lymphoids that generally associated with mucosa, for one. So nodules with mucosa. And they're also called MALT, which is called mucosa-associated lymphoid tissue. Lymphoid nodules are different due to location organization. They have no capsule, no afferent vessels, found in areolar tissue, in mucous membranes, density high where there is much pathogens. For example, in the colon, you see a lot of lymphoid nodules in the form of MALT, and they contain both B and T cells. Another thing is the spleen. The spleen contains a lot of lymphoid tissue that monitors blood, filters and monitors blood. It also has non-lymphatic functions such as recycling old red blood cells in the red pupils. And white pupils, they have white pulps, not white, like red pupils, red pulps. So I guess I'll write it here. Red pulps is where recycled old red blood cells, and the white pulp is where there's clusters of lymphocytes. And the spleen filters plasma, not lymph. However, the spleen is has a capsule and is classified as an organ, but the organization of lymphocyte clusters is much more similar to that of a tissue. So these are big players in the lymphatic system. You just have to know the differences between them and whatnot. And now for inflammation and innate immunity. Sorry, let me just see something. So innate immunity um, is present from birth and is the same for every one of the threats, whereas adaptive would be learned and much more specific. Now, cellular mediated immunity contains first two factors that are very key. One is a complement, and these are plasma proteins that lead to antimicrobial defenses. And they also have cytokines, which are any signaling molecules for cell-mediated immune defense. And the basic components, remember, are recognition, recruitment, and resolution. Now, give me a second to catch my breath. Okay, I need to organize this. Okay, so innate immune responses can be called inflammatory immune responses and have two phases. So innate, we have phase one, the immediate response, and two is the induced response. And in the innate, it's generated by soluble by cells and tissues within the local tissue, within local tissue, cells and factors within local tissue, as the induced response is recruitment of white blood cells from blood circulation. There's five signs of inflammation. One is heat, other is redness, other is pain, swelling, and then loss of function. I think the best idea to imagine this is to imagine like a blushing rat, a blushing fat rat. So why it's blushing is because it's hot. It's in pain, likely because it's hot, because you injected it a lot of stuff from a lab. It's going to be swelling, likely, so pain, hot, swelling. It's going to be red, probably, because you're making a blush. And it has loss of function because you gave it a loss of function drug. So first, resident white. So primarily resident white blood cells. I think I should call them resident equals mast cells. This is an induced response more so. They recognize damage, and they also recognize pathogen entry. And so these mast cells, they secrete histamines and hesperins from mast cells. The mast cells degranulate. They release histamines, hesperins, and cytokines. And this leads to changes within the local tissue. Well, first, it attracts more white blood cells to enter, white blood cells to come. It leads to capillary widening. So it's going to get wider like this. So I guess this goes into our idea of redness and heat. Redness and heat are associated with capillary widening. And it's also increased permeability. So this is fluid release into tissues. So that's going to be fluid release into tissues. That's going to be swelling. And yeah, it also leads to attraction of leukocytes, the white blood cells entering, and then the systematic response of proliferation and uh, which is going to be pain. You're going to have a lot of stuff entering. It's going to be kind of painful. And then it's going to also lead to loss of function, meaning a systematic response. And then in terms of cytokines, we mentioned here for brief, one type of interleukines kind of generate a lot of inflammatory responses. So IL-1 can trigger fevers by changing hypothalamic set point or temperature. And then we also have interleukin 6, yeah, which can lead to proliferation of white blood cells in red bone marrow, specifically neutrophils. And then releasing of factors through mast cells leads to, again, this recruitment of, well, this is a symptom of fever. So I guess that leads to swelling. It can also lead to pain. Fever can be leading to pain. Loss of function can definitely be leading to IL-6, or secreting all these cells. And releasing of these factors from mast cells leads to recruitment of circulating white blood cells through a process called extravasation. Extravasation, so mast cells secreting all these cytokines leads to extravasation. Extravasation basically just means leukocytes entering the space, basically white blood cells entering tissue, and that's going to be pain. And then extravasation includes changes to endothelial wall, allowing white blood cells to enter, and then hemostasis of white blood cells into space, meaning it's going to move it. So I guess we can kind of just, um, well, I don't want to summarize it or anything, but first I want to take a step back. First, we have these mast cells, and these mast cells, they secrete histamine, cytokines, and heparin from what's it called? Not, yeah, mast cells secrete histamines. Uh, they degranulate, secreting histamines, hesperins, and heparins, and cytokines. And cytokines can include interleukin 1, which leads to fevers by changing the hypothalamic set point temperatures, and also interleukin 6, which can induce white blood cell production. And then with white blood cell production, proliferation, I should say, in red bone marrow, this leads to release of factors of some factors from mast cells can lead to recruitment of circulating white blood cells through extravasation, extra, extra, ex, extravasation, and through extravasation, this changes the endothelial wall to allow white blood cells to stick to it and enter. And also, this allows for chemostasis of white blood cells into the space. Now, another type is including phagocytic myeloid cells. They just basically eat up pathogen. And these are using a macronym M. This is called macrophage, eosinophils, neutrophils. They directly consume it. And they have different anatomical and physiological profiles and are used for different responses. So macrophages can be either fixed or free, and their removal of dead cell debris. Eosinophils are usually pathogens that are targeted by antibodies, and neutrophils are abundant and the main type recruited during innate immune responses. Now, pus specifically, if you ever had an injury, you have like some sort of white infected area. This is a protein-rich fluid with dead leukocyls, mostly neutrophils, at a site of infection, and this is due to wear and tear, certain bacteria or certain like toxins from certain bacteria that kill white blood cell, and it's common for bacterial infections other than other type of infections. Complement proteins are also soluble immune factors that assist in resolving bacterial infections when activated. So I believe, did we mention? Yeah, we have either cytokines and we have complement proteins. Complement proteins are for microbials. Complement proteins are for microbes, like more. And they're made in the liver to the blood plasma. And this leads to a cascade. So the activation complement 3 splits into 3 complement 3A, complement 3B. Complement 3A leads to inflammation and activates, which means it's activating mast cells. And complement 3B triggers other complement proteins to kill cells. So cell lysis. And this uses something called the membrane attack complex. This can also bind to bacterial surface and increase the efficiency of phagocytosis, which is called opsonization. So membrane attack complex, MAC, destroys cell wall. Help destroy the cell. So again, there's CA, which basically is inflammation, which is recruitment of activation more mast cells. There's CB, which can either aid in phagocytosing it using binding onto the bacterial surface, or they can aid in direct cell lysis through, and they can recruit other complement proteins that trigger cell lysis, or they can basically just destroy the integrity of the target cell wall using membrane attack complex. And these can be activated in 3 different ways. So 3C can be first in the classical pathway using antibodies, in the lectin pathway, which is using the bacterial cell wall, interacting with the bacterial cell wall, and the alternative pathway, which is stabilized by complement factor P, stabilizes C3b. So C3b is secreted by, I should note, secreted by bacterial stimulations or stimulated by bacterial secretions. The classical pathway is through antibodies. The lectin pathway is through the bacterial wall, interaction with the bacterial wall, and 3 is with complement factor P. And yeah, remember the complement system is from mast cells or column or recognition, I should say. Recognition is from the mast cells or complement systems. Recruitment include key cytokines, vasodilation, permeability, chemostasis, and extravasation of white blood cells. And the resolution is either phagocytosis or lysis by complement membrane attack complex. Natural killer cells are also another cell. They provide surveillance, destroy abnormal cells. An abnormal cell is either infected by a virus, or part of tumor. So how they do this is identify unusual cell surfaces, or they secrete a cytokine called interferon alpha, IFN alpha. And this locates abnormal cells. And this releases substances that trigger cell membrane permeability and lysis. So cell membrane permeability plus lysis. And they can also secrete tumor neurotic factor alpha, which acts as a peregrine that impops apoptosis in tumor cells. So IFN alpha is a cytokine, whereas TNF alpha is just apoptosis. Basically, IFN alpha attracts phagocytes to remove debris. And IFN gamma is a chemocyte attracting macrophages to the area. And this is the steps of innate immunity. So as for adaptive immunity, main key factor is adaptive immunity is used when innate immunity is unable to specifically remove the pathogen. And it's different because it's more specific, it has memory, it has versatility, and it has tolerance. So for each of these, let's say specificity, they have receptors to identify specific antigens, ignoring others. As memory, they can divide, it's basically activated. B or T lymphocytes can divide into memory cells, mounting a stronger future attack to same antigen exposures. They can be more versatile, versatility. They can recognize a diverse set of antigens, and they can also be tolerant. And they ensure self tissue is not a threat, whereas non-self is a threat. Now, the big players is that antigens are molecules that perform the role. T cells use cell-mediated immunity, while B cells use humoral immunity. So basically, T cells use other cells to attack, whereas B cells use antibodies that target it for destruction. So target for destruction. It's basically like a bomb rather than attacking it directly. So primary lymphoid structures are bone marrow and thymus. And the thymus is special because T cell maturation and selection is in the thymus. So lymphocytes determine the target using three complexes, MHC1 and MHC2. They only identify antigens when they're bound to molecules. And when an antigen is presented to an MH molecule, they learn to direct it. Let's say it's like a freaking person with no direction at all. If you give them a recipe, let's say MH1, they're going to say, okay, we're going to go towards MH1 and we're going to do this. Whereas if you give them MH2 or something, they're going to do that instead. It's think of it like a these MH complexes as a specific target that they want to look for. Maybe it's like they're trying to hunt someone down and you give a bounty to them and you have a sort of photo of the person, kind of like, yeah, like a wanted poster. And intracellular targets are presented on MH class ones. So intracellular, let's just write this out first. Where antigens are MH1 and they're found in all nucleated cells. So intracellular antigens will be from viral or bacterial infection. And then extracellular antigens are phagocytized and then for MH2, they're on the surface of antigen presenting cells and lymphocytes, and they're extracellular. So remember, one is for nucleated, I think one is nuke, and two is antigen presenting cells. And these would include dendritic cells, macrophages, B cells, and then the mast cells, basophils, and whatnot. And the only ones that I would say MH1 can't be on is for unnucleated cells. So I think only maybe I'd say platelets and what's it called? Platelets and red blood cells don't have any nucleus, so they would likely not have any of these functions. But yeah, that's how it is. T cell antigen relies on antigen presentation. Here's antigen presentation by MH1. And yeah, MHC molecules are recognized onto T cell surfaces, and they're unique to each lymphocyte. Different lymphocytes recognize different antigens. And yeah, different T cells recognize different MHC molecules. And there's different sorts of differential clusters that could be on the surface of, let's say we have a leukocyte or a lymphocyte here. There's different these sort of clusters, CD markers. And CD markers play a role in antigen identification and whatnot, but you just need to know CD4 is, is in MHC1, which is in T cells. CD8, oh wait, I should say it's an MH, oh God, CD8 is an MH1. And CD4 is an MHC2, which is in B cells. So the rule is 8. You see 8 times 1 is 8, and 4 times 2 is 8 as well. So these are the CD markers for these sort of MHC cells. T cell response is guided by three signals. So first, the MHC interacts with T cell receptor. And then two, we have a co-stimulatory molecule. And three is a cytokine. And all these can be both pro-inflammatory or anti-inflammatory. And this is determined on two or three. So first you have the MHC needing to combine with the T cell receptor, and then you just need to know two C's. One is co-stimulatory molecule, and the other one is cytokines. Cytokines are kind of in everything, so I guess you kind of just need to know one C in this aspect as well, either. So now antigen recognition and whatnot and simulation also leads to CD8 T cell activation. Sorry, give me a second. Oh, my back is hurting. Oh, I guess these are all MHC1 and MHC2 are all recognized by T cells. This is basically all the T cell stuff. And basically antigen recognition and co-stimulation leads to T cell CD8 T cell activation when they're presented onto MHC class 1 molecules. And when MHC1 molecules, they become primed. And for a full, so for CD8 plus T cell, they first have to have an antigen attached, and then this will become primed. Upon being primed, it needs to be co-stimulated from infected cell or surroundings. And this activates T cell division and differentiation. And the activated CD8s become activated CD8s become cytotoxic cells or memory T cells. So cytotoxic or memory T cells. T cells, cytotoxic T cells, they directly do the work. They release cytotoxins that lead to cell death onto the antigen-bearing cells, whereas memory T cells are inactive in the secondary lymphoid organs until later infections. I remember at activated CD4 cells, so CD4 complex plus T cell, they differentiate into active helper T cells, memory T cells. Oh, I should say this is TC cells for all these. And then, or regulatory T cells. So after being primed and presented with MHC2, it also needs the co-stimulation, or as co-stimulation from signal from the infected cell, or from the environment, co-stimulation from infected cell or environment, and then it creates cytotoxic T cells, memory T cells, and helper T cells. So regulatory T cells, they suppress immune response, so it creates inhibitory cytokines. Memory T cells and cytotoxic and regulatory T cells kind of just regulatory T cells they inhibit. Helper T cells secrete cytokines. I'll mention that later, I guess. Cytokines. And then memory T cells do the same. They just stay in the secondary lymphoid organs until a later exposure. And again, cytotoxic T cells, they induce cell death on the target cells. So remember, CD8 secretes TC cytotoxic and memory cells. Memory cells stay in secondary lymphatic system and lymphoid organs, and the cytotoxic ones induce cell death. They need to undergo an activation and become primed and then co-stimulated from the infected cell or from its surroundings. The response is guided from the MHC interactions, co-stimulatory, co-stimulation, and also cytokines. So again, yeah. And then this sensitization precedes B cell activation. So B cells attach onto bind antigens and become sensitization. And once it's sensitized, it requires the helper T cells to come here and activate it. And B cells, they phagocytose, they endocytose antigens, present them onto MHC2, and presents onto cell surface. So they would use a CD4 markers. And then upon activation with CD4 ideas from helper T cells that represents the same antigen, they become activated. And upon activation, they divide and differentiate. They come into active C cells. They have memory B cells, which are inactive. And the active B cells turn to the plasma cells. This is directed by cytokines. Again, the inactive memory B cells will stay in secondary lymphoid organs until subsequent antigen exposure, of which will turn into plasma cells and secrete antibodies. And the plasma cells can secrete antibodies, yeah, specifically. So what necessarily are antibodies? Antibodies are small proteins. You have a variable chain. You have these variable segments. And then you have these constant segments here. Kind of look like this. And when they bind, and the variable segments are able to bind to target antigens. The part of the constant segments are accessible and they bend to complement proteins or innate immune cells. So complement proteins or innate immune cells. And these antibodies use a variety of mechanisms to defend the body from pathogens. They can neutralize and prevent adhesion. They can block pathogens from binding to healthy cells. They can be a part of complement systems, bind complement proteins. This tags the antigen for degradation, and they can also do opsonization, phagocyte traction, and inflammation. They engulf pathogen and release pro-inflammatory cytokines. There's also 5 different classes of antibodies called immunoglobulins. So we have IgG, IgA, IgM, IgE, and IgD. These are part of B cells and bind to during sensitization. Binds to sensitization means binding to antigens. E cells are part of allergic reactions, binds to mast cells and basophils. This is the first type to be released, circulates around blood and lymph. A is part of glandular secretions and mucous membranes to prevent pathogens from accessing, so blocking pathogens. And G is for 80% of antibodies. So G is a general one, N is the first one as secreted in the blood and plasma, blood and lymph. A is for glandular secretion and is for basically just neutralization and preventing adhesion. I should say neutralization and preventing adhesion somewhere here. The complements are tagging it for degradation, which is basically D in a way, targeted for degradation, and phagocyte attraction would be IgE, phagocyte attraction and inflammation. And adaptive immunities improves during the subsequent infection. So first you'll see IgM generally, and let's say we'll have another Ig whatever. Let's say we could call it IgA, IgG, whatever. And then this is the first time. The second time they see it, they're going to have a much subsequent response. IgM is still going to have a regular response, but in terms of the memory T cells, IgG is going to have a uh, a titra that suppresses the primary response, both level and time. So antigen titers generally peak 1 or 2 weeks after the antigen exposure, so you should see it around here-ish that peaks it. And resolution is basically done through innate and adaptive immunity cells. So let's say we have an onset of an infection. Well, we're going to have a lot of cells recruiting, so onset, and then resolving it, we're going to not need that many. Resolution, post-resolution, we also need a lot. Why? Because we need to restore the function. So first, in terms of resolution, you remove all pro-inflammatory cytokines. And immune cells clear the area of inflammation and undergo apoptosis. So immune cells clear, undergo apoptosis. This is similar to removing pro-inflammatory cytokines. And this is removing everything related to inflammation and post-resolution. We have influx of regulatory T cells to establish immune suppression and tissue resident mac macrophages and dendritic cells, they return. So yeah, this is essentially just everything within the immune system. I guess you can also talk about measles versus adaptive response. Measles are coming from the virus. They infect the in alveolar macrophages or dendritic cells. And yeah, it's just some case study stuff. Yeah, this is basically the big ideas of immune system. Next time you're going to hear from me, we're probably going to be going over digestive system. Thank you for listening.